Good day everyone, I am Desert and Francisco, and together with me are Mr. Bibera and Mr. Olimpayan, and for this part of our presentation, I'll be discussing the protein extraction from corn kernels, scientifically known as zea mays. For the procedures and materials, the extraction from protein from zea mays, we started by grinding 50 grams of corn grains using a mechanical blender. We then added 125 ml of distilled water and macerated the mixture for 4 hours to help release the protein content. Afterward, we filtered it using muslin cloth to separate the solid parts and then passed the filtrate through filter paper. The collected filtrate was used for different confirmatory protein tests later on. In this slide, we documented each step of the process. We first separated the corn kernels weighing a total of 50 grams. We then homogenized the kernels with enough water, macerated them for 4 hours, and filtered the mixture. We collected 85.6 ml of clear filtrate, which served as our sample for confirmatory protein testing. In terms of organoleptic properties, the extract proteins varied slightly. The protein soluble in water and the globulin appeared both as clear liquids, and prolamine zane showed a clear liquid with a hint of white, slightly slimy or jelly-like in texture, while the glutamine remained clear and liquid. These characteristics were actually observed in the laboratory during the testing. For the solubility test, we tested each protein in water, alcohol, and ether. The protein soluble in water was found to be soluble in water but insoluble in alcohol and ether. The globulin and glutamine were insoluble in all three solvents, while prolamine zane was insoluble in water but soluble in alcohol. For the chemical identification, we performed the Santaproteic test for protein. These tests reacts with aromatic amino acid to produce a yellow color. In our results, the protein soluble in water, globulin, and glutamine each produce a yellow color liquid, while the prolamine zane produced a light yellow color, and these changes confirm the presence of proteins in our corn extract.
So moving on to the questions to answers part. Number 1, how are proteins classified on the basis of their chemical nature and solubility? And give example on each classification. So proteins are classified based on their chemical nature and solubility into simple and conjugated types. So simple proteins consist only of amino acids and differ in solubility. So for instance, albumin are water soluble, while keratin and collagen are insoluble structural proteins. So conjugated proteins contains a nonpolar prosthetic group such as carbohydrates in glycoproteins or pigment in chromoprotein. Number 2, in Chantropic test, what is the mechanism of action in the formation of yellow precipitate as an indication of protein? So in Chantropic test, a yellow color precipitate indicates the presence of aromatic amino acids such as tyrosine, tryptophan, and phenylalanine. So concentrated nitric acid reacts with their benzene rings through nitration, forming yellow nitro compounds. So when an alkali like sodium hydroxide is added, the sodium turns orange, confirming the presence of aromatic groups in the protein. Number 3, give the composition of Million's reagent. So the Million's reagent is composed of mercury and nitric acid. It is prepared by dissolving metallic mercury in concentrated nitric acid and then diluting it with water. So the main active component is mercuric nitrate, usually mixed with nitrous acid formed during the reaction. So this reagent is used to detect tyrosine proteins, which gives a white precipitate that turns red upon heating.
The presence of protein in corn was effectively validated through sensory evaluation, solubility assessments, and chemical testing. All methods produce positive outcomes, confirming that proteins are present in the corn sample. The yellow color noted in the Xanthroprotic test further sustained the existence of aromatic amino acids, affirming the various types of corn proteins, albumins, globulins, prolamine, and glutenin are all identified. In summary, the experiment successfully illustrates the identification and confirmation of proteins in corn.